So I've had questions like I explained cleavage and I mentioned something like cell division. What is cell division itself? So let me just quickly explain that. You see, cell division is a process, a biological process whereby a parent cell divides to form daughter cells, either two or four daughter cells, and I'll be explaining how it can be two and how it can be four. You see, cell division is just a part, a fraction of the larger part of the cell cycle, where cell growth, replication of the chromosome is also part of the cell cycle as well. Now, based on different criteria, cell division can be two in eukaryotes because we have two type of living organism based on the type of cell that they have. We have the prokaryotes and we have the eukaryotes. Prokaryotes are those ones that have no true nucleus, and eukaryotes are the one with true nucleus, right? Now, in eukaryotes, we have two type of cell division. We have this vegetative cell division, which is known as the mitosis. It's also known as somatic cell division. That is this division for growth and development. And we have the division for sexual reproduction, that is meiosis, that occurs either in the sperm cell or the egg cell, and it is also known as reduction division. So I can say it is either somatic cell division or reduction cell division. And the somatic cell division, we have the diploid, in which the daughter cells that is produced is genetically identical to the parents, which means if the parent has 46 chromosome, the daughter cells will also have 36 chromosome. And for the reduction division, it is different, you understand, from the parent cell. If the parent has 46 chromosome, the daughter cell will have 23 chromosomes. So that is just it. That's why it is called the reduction division. So in mitosis, which is vegetative division, and a diploid cell is formed, and in meiosis, an haploid cell is formed. Now, you see, prokaryotes, they use a special vegetative division known as what? Known as binary fission. And this binary fission, we have three processes. The first one is DNA replication. The second one is chromosome segregation, and the third one is cytokinesis. Now, basically, in eukaryotes, the eukaryotes as well, we have two stages of cell division. We have the karyokinesis, that is division of the nucleus, and cytokinesis, which is the division of the cytoplasm. You understand? Now, in cell cycle itself, cell cycle consists of different stages. We have the interphase stage, the mitosis stage, the G1 phase, G2 phase, synthetic phase, which is the S phase. And basically, in mitosis, I said mitosis, we have diploid cell will be produced, and meiosis and haploid cell will be produced, right? So basically, in cell division, we have different phases. You understand? Like I said, cell division is just a fraction in cell cycle. So we have different phases, and part of the phase that we have the it is acronymed IPMAT. IPMAT. That is the interphase, prophase, metaphase, anaphase, and telophase. You see these five phases, they are the karyokinesis. That's the division of the nucleus. You understand? Where there is DNA application, chromosome segregation, and all like that. So basically, the interphase is known as the resting phase, and it takes the larger part of the cell cycle itself. You understand? Now, in the prophase, we'll be seeing the appearance of chromatin network, which become condensed at the end of the prophase, and at the metaphase we'll be seeing the chromosome forming and all like that. And at the end of the telophase, we would have seen that the nucleus would have divided completely. So after the karyokinesis, we now have cytokinesis. That's the division of the cytoplasm itself. You understand? So the cytoplasm is going to divide into two cells. So we are going to have two daughter cells, and if it is meiosis, we are going to have meiotic 1, meiotic 2. And in meiotic 1, we have the prophase. The prophase stage, which is the longest phase in the meiotic 1. We have, I think, 5. That is LPZD squared. Now, and that is leptonema, pachinema, diakinesis, diplonema, and zygonema or zygoteny. So we have them like that. That is there are the stages in prophase. You understand? The prophase. So after the prophase 1, after the meiotic 1, where we have prophase 1, metaphase 1, anaphase 1, we move to what? We move to telophase 1. We move to the prophase 2. You understand? That's the meiotic 2. So that is what we have there. And the cell is going to divide into basically two daughter cells. You understand? Or if it is meiosis, that is four daughter cells, which are haploid in nature. Now I'm talking about chromosome. You see, chromosome is where the DNA is stored, or the genetic material is stored itself, because in meiosis, it is the division for reproductive system, like the reproductive cells, either sperm cell or egg cell, especially in humans. Let me use humans as an example. If you watch one of the videos that I've made before, I explained the histology of male reproductive system, and I was talking about the formation of sperm, right? And if you check it very well, I explain what meiosis is there, where we have one parent cell that divides into four daughter cells. You understand? The spermatocytes become spermatozoon. So we have them like that. You understand? So and this is what cell division is. Now, how does it relate with cleavage? Cleavage is also a type of cell division. You understand? So a type of cell division, but in cleavage, cytokinesis does not occur. Like the cytoplasm does not divide. Every of the cells, they divide inside the cells, inside the main cell. Now they divide there and they remain there to form a cluster of cells, you understand, known as the morula. And the morula develop into the blastula, you understand. And we have different. So after blastulation, we have gastrulation as well, which I'll be explaining. So this video is just basically to explain the basic concept of cell division. That is what I just made the video for. Thank you for listening.